I will present to you today something about a part of our results dealing with the interaction between proteostasis and metabolism. We came across this story because everybody here, I guess, is familiar with the hallmarks of aging. Briefly, they are a group of phenotypes that arise during aging that also will extend the lifespan of the species if ameliorated, or will accelerate aging if aggravated. This is briefly, let's say, what one can say about the hallmarks of aging, and they are more or less conserved across the species. What we are interested in is the interaction between the different hallmarks of aging, and these are loss of proteostasis, deregulated nutrient signaling, and mitochondrial dysfunction. So we are trying to study their connections and not only each of them in isolation. Our main research interest, and I would say also still somewhat hypothetical, is that RNA and protein folding homeostasis can reprogram cellular metabolic activity, which will then, in a way that we have yet to describe, feed back towards the RNA and folding and protein folding homeostasis. What we can say already is that their communication is what is required for the maintenance of cellular homeostasis. We are using budding yeast as a model organism. We are measuring the replicative lifespan in budding yeast, which is defined as the total number of cell divisions per mother cell. As you can see in this picture, the mother cells are much bigger than the daughter cells, and they are easy to distinguish when looking under the microscope in this little device called the micromanipulator, where we basically organize the virgin mother cells in an array, and as they undergo divisions, we remove the daughters one by one and make a note until all the mother cells stop dividing. So today, briefly, the message that I will try to convey is about the macromolecular homeostasis modulating the metabolism of the cell, which can then in return support the macromolecular homeostasis. First, I will tell you briefly how we came to find this crosstalk and how the interaction, although not necessarily a direct, a physical one, between HSP90 and TORC1 is mediating this crosstalk. So we were kind of, let's say, inspired by the result of several groups that the wild type cells will usually have approximately 20% of proteins targeted to degradation immediately after their synthesis. And we were wondering what will happen if we decrease this percentage, if we manage to make the protein folding, the status of the proteostasis better, if we improve it. And you can find the details in the paper. Also, I will be happy to discuss afterwards. But this kind of manipulation, so we overexpress different chaperones from different compartments, and it all converged. They all converged at the downregulation of HSP90, which led to the inhibition of TORC1 complex. This was then found to lead to the activation of the SNF1 kinase, which is a yeast homologue of the AMPK, mammalian AMPK. Activating SNF1, the response to this brought to a response that is a phenocopy of the glucose starvation response. Of course, we didn't know immediately how important this signaling cascade is. So our replicative lifespan measurements are basically suggesting that while in different, I'm just showing two here, we have done four that we report about in the paper. But while in the wild type background, in the control background, we see a lifespan extension. If we overexpress the same chaperones in a background that cannot deactivate TORC1, that has a constitutively active TOR1 kinase, there is no lifespan extension anymore. So the same if we delete the SNF1 AMPK, and the same if we make the yeast respiratory deficient. So if we abrogate this communication in either of these positions marked with red arrows, there is no more, we cannot see anymore the replicative lifespan extension, which would suggest that this crosstalk is actually in a way required for this improved proteostasis to lead to the lifespan extension. The next thing, we were curious, what would then happen during heat shock? Heat shock induced lifespan extension is something that is well known that it happens, but and also across species, but then we didn't know the mechanism of this. And then, inspired by this data, we were wondering if we can find something similar also there. So first, what we did is we subjected the cells to heat shock. Since yeast optimally lives at 30 degrees Celsius, we put them for 3 hours at 34 degrees while the cells were still very young. So they were 1 to 3 generations old. And we found this lifespan extension. What we also found, though, was that there is a strong increase in oxygen consumption. So I have to say that these cells were heat shocked during their exponential growth, when yeast cells are known to rely on glycolysis because of the abundance of glucose. However, these cells, when they are subjected to heat shock, they seem to switch to respiratory metabolism. And then, inspired by the previous results, we thought, okay, maybe respiration has something to do with this as well. So what we did is we included in the study 2 mutants. One is, again, the respiration deficient mutant, the petite mutant, which does not respire in optimal or heat shock conditions. But we also included another mutant with the HUB4 transcription factor deleted. This transcription factor is regulating the switch to respiratory metabolism. So while in optimal conditions this strain can respire, it cannot switch to respiratory metabolism during heat shock. And then we measured their lifespan and found that in none of the cases heat shock extends the lifespan. What else is happening during heat shock? We found that together with the increase in oxygen consumption, there is an increase in the levels of mitochondrial superoxide and, of course, also ATP. And then at the transcript level, we found something that caught our attention immediately, which is activation of antioxidant protection in terms of superoxide dismutases, catalases, and glutathione reductase, as well as an activation of the G6PD, which is a rate-limiting enzyme of the pentose phosphate pathway. So it seemed that these, of course, in addition to activating the heat shock response, these cells are under oxidative stress and they're trying to fight it, to fight against it. And the only question we were wondering about is why then the pentose phosphate pathway. This is a very nice story, by the way. We collaborated with Nuno, who is talking after me, and actually he suggested that this is very logical, since glutathione recycling, that is a part of the antioxidant defenses, require glutathione reductase, which is this enzyme here, which then is using NADPH for the recycling itself. NADPH is one of the main products of the pentose phosphate pathway. So what we did next, there are many more details again that we reported in the paper. I'm trying to just show the main things here. What we tried next is to neutralize the mitochondrial superoxide. So we overexpressed the mitochondrial superoxide dismutase, and we confirmed that this strain will also undergo an increase in oxygen consumption. However, it does not activate antioxidant protection or the pentose phosphate pathway. Finally, in the absence of superoxide, with superoxide being neutralized, there is no replicative lifespan extension either. We also wanted to see what happens with TORC1 in this case, and we found it also to be deactivated during heat shock. We used two methods here because the antibodies to measure the phosphorylation of downstream targets of TORC1 for yeast, they are a disaster. So we are we set up two different methods. One is to measure the transcript levels of several genes that TORC1 acts as a transcription repressor. So when TORC1 is deactivated, their levels will increase. We have rapamycin here for control. And then we also set up an imaging assay where we had TOR1 tagged with GFP and analyzed the colocalization with the vacuole. So while in the optimal growth temperature, you can see that TOR1 kinase is colocalizing with the vacuole. In heat shock as well as with rapamycin, you can see that it's basically forming some sort of speckles in the cytosol. And then again, if we heat shock the strain that has constitutively active TOR1 kinase that cannot deactivate it during heat shock, again, this strain does not experience the lifespan extension, despite the fact that they're all activating the heat shock response. So this is something, a more direct evidence is not possible because the deletion of HSF1 in yeast is lethal. But what I can say that at least there is a lack of correlation between the activation of the heat shock response and the lifespan extension. So all of these strains that show no lifespan extension whatsoever in heat shock, these different mutants, they all activate the heat shock response similarly. So this is how I would summarize this part. We don't know exactly in which way TORC1 gets deactivated in heat shock. This is something that we're working on. Again, we are hypothesizing that there must be some story with certain chaperones, but in this moment we don't know. But in any case, something that starts with an increase in ROS levels culminates in the lifespan extension and does not seem to have a direct connection to the chaperone levels. And finally, we were wondering what is happening with, what would happen in the opposite direction. If we create a metabolic stress, what would happen to proteostasis, specifically to protein solubility? And then which better way, at least to our knowledge at the time, which better way to trigger a metabolic stress than to bring down the ATP levels? So what we decided to do is perform glucose starvation. We had another motivation here. So I'm not sure how many people here are familiar with different types of protein aggregates in yeast, but very briefly, and also only for the schematic purposes, a certain type of aggregates called Q bodies are nucleated. So for their formation, they require the chaperone HSP42 which is going to build this type of aggregates. What pulls the proteins out is HSP104 disaggregates, so it's going to pull out the proteins not only out of Q bodies, but also it's going to work as a disaggregate of all the different aggregate types in yeast. And for pulling out only one single protein out of the aggregate, it requires 20 ATP molecules. So we decided this to be our model, let's say, while studying the effect of metabolic stress on proteostasis. So what you can see here is two different regimes of starvation, a mild one, 0.2% glucose, and a more severe one with 0.02% glucose. And always on the left of each of these graphs, you can see how many, so you can see the percentage of cells with one aggregate, and then on the right, percentage of cells with two or more aggregates. So you can always see how the starvation is increasing the aggregation, the aggregate abundance in these cells. But if during the last 30 minutes of the 90-minute starvation regime, we introduce also ATP, then my arrow is lost. I cannot point actually in this. Here it is. So if we add ATP into the medium, you see that also here you can see that the aggregate abundance is decreasing. The same effect we do not have with GTP or ADP. And then here you can see the similar effect to aggregate sizes. So we measured the diameters of these aggregates, and then adding ATP to in both starvation regimes is leading to a decline in the diameters of aggregates down to the control level. Again, GTP and ADP do not have this effect. Then we created two mutants because there was a report, of course, from Anthony Hyman's lab that ATP alone can do these things. It can fight aggregation. We wanted to check if this is also the case in our system. So we created two mutants of HSP104 that can bind but not hydrolyze ATP. We targeted two different nucleotide binding domains, and basically any of them. So while adding ATP to any of these mutants, it didn't help. It did not dissolve. So they were not able to dissolve the aggregates. So ATP hydrolysis activity is required for this. You can see that the similar effect we saw in for each of these mutants. So this is the NBD1 mutant and NBD2 mutant. Adding ATP did not help in any of the starvation regimes. What is pointed here with the red arrows is the vacuolar localization of the signal. And of course, structures like this were not taken into account in this size quantification. What I also want to point out here, what I think is one of the main messages of this work, is that already in optimal conditions, we see aggregates of larger sizes than in the control in the previous slide, right? So what this is telling us is that there is always stuff around that can be loaded into the aggregates. But so even in the most optimal conditions, there is stuff that can be put there. But what matters, it seems, what is going to determine the final size and the abundance of these aggregates will be how efficiently they are pulled out of there, at least in yeast, right? We still don't know exactly what's happening in mammalian cells in this context. I will quickly show you also how this looks like, what this looks like kinetically. So if you look at the plot here on the left, here in the moment zero is the beginning of starvation. So you can see how quickly the ATP levels are going down and how growth of the aggregates is corresponding here. At the same, and they reach some sort of saturation approximately at the same time. On the other hand, here you have at point 0, you have the beginning of recovery. This is when glucose is given back to the cells, and you can see how the ATP levels are restored and how the median diameter of aggregates is decreasing. In the mutant, while the assembly of aggregates is following almost the same kinetics, you can see that during the recovery, while ATP levels are recovering, there is no dissolution of the aggregates. To summarize this part, we found that there are 2 types of aggregates being formed there, which is Q bodies and stress granules, and that they are accommodating misfolded proteins and nascent polypeptides. In the interest of time, I'm going to only say that we were able to determine that the ATP that is required for the dissolution is coming from glycolysis, and that reactivation of glycolysis that depends on the pKa post-starvation reactivation was an essential event for this entire business to take place. So both for ATP levels recovery and for the dissolution of aggregates. And this brings me to this schematic that is basically a relationship between proteostasis and metabolic stress that we were able to describe through this work during glucose starvation. But what it inspired us sort of is whether this is a relationship that we're looking at also in aging, whether the proteostasis failure is indeed an independent event during aging, or it is something that is a consequence of strong changes in metabolism in cells. And from the conclusions, I just want to point out one of them, which is that if this is something that is as relevant as it seems to us in this moment, it would be worth to describe what we call right now proteometabotypes, which would be certain combinations of protein folding quality and metabolic activity in different types of cells. And then, of course, this would be, let's say, a very long-term aim of this project, which would also then allow us to recognize failures in metabolism to respond to the needs of protein folding, and perhaps this would tell us something new about the protein folding diseases, or even aging or cancer or something else. And then in the end, I don't know how much I'm over time. I just want to thank my colleagues from Mediterranean Institute for Life Sciences that have been working with me on this very hard until a year ago that I moved to Göttingen. And I want to thank my collaborators, my former and current collaborators at University Medical Center in Göttingen. This work was strongly supported by Nuno Raimundo and Ira Milosevic. And thank you very much for your attention.